Good evening, everyone, and welcome to LSU Science Café. Dr. Steve Beck is not able to be here with us tonight, but I'm Allison Satake with the LSU Office of Research and Economic Development. And we are excited to bring you tonight's program featuring marine biologists, Dr. Morgan Kelly and Dr. Jacqueline Padilla-Gamiño, who will be speaking on how climate change is affecting life in the ocean and which species are likely to be the most vulnerable. Live closed captioning for this event is available. Click on the live transcript button at the bottom of your screen. Despite our physical distance, our goal is to continue to build a strong, informed community by providing access to reliable information, new ideas, and cutting-edge research, scholarship, and creativity from faculty experts and researchers in fields across the university. Tonight's event is brought to you in partnership with public radio station WRKF, based here in Baton Rouge. WRKF will be giving away one stainless steel NPR Science Friday beverage tumbler to one lucky audience member tonight. To enter the WRKF drawing, click on the link in the chat button and fill out the Google form. Okay, it's now here in the chat button. Fill out the Google form with your contact information. Also, please be aware that this online event is being recorded. It will be available with a transcription from the LSU Research website within the next few days. And now, without further ado, I'd like to introduce tonight's speakers. Dr. Morgan Kelly is an associate professor in the LSU Department of Biological Sciences. Her research focuses on the ecology, evolution, and conservation of marine invertebrates, including oysters, in coastal Louisiana. She is an LSU rainmaker and a recipient of the prestigious Alfred P. Sloan Foundation Research Fellowship. She is joined by Dr. Jacqueline Padilla-Gamiño, who is an associate professor in the School of Aquatic and Fisheries Sciences at the University of Washington in Seattle. Her research focuses on how climate change will affect the ecology and reproduction of marine invertebrates and seaweed. She is also a recipient of the prestigious Alfred P. Sloan Foundation Research Fellowship. After the presentations, we'll have a moderated question and answer session. To participate, please use the Q&A button to enter your question at the bottom of your screen. We'll be monitoring that section and relaying them to our panelists. So now I'll turn it over to Dr. Morgan Kelly.
Hi, everyone. Thank you for being here tonight. It's really nice to see faces and names in the chat, at least. I want to say thank you to Allison and to the Office of Research for organizing this event. I'd like to start our conversation with this photo. This is a photograph taken by the Apollo 8 mission during lunar orbit. And it's been described as the most influential environmental photograph ever taken. And that's because when we look at our planet from this distance, we can see that it's small in this vast expanse of space. And it's the only place we know that can support human life. We can see that we share one planet with limited resources. But as a marine biologist, I'd also like to point out from this photo that most of the Earth is blue. The oceans cover 70% of our planet, and they provide critical resources to us humans. They provide 17% of the animal protein we eat. They provide homes for 60% of all people live on a coastline. And the oceans provide about half of our planet's oxygen. About half of our atmosphere's oxygen is produced by tiny little algae called phytoplankton. And so you can thank the ocean for every second breath you take of oxygen. At the same time, ocean life is being threatened by changes caused by humans. The oceans have absorbed 90% of the heat that has been trapped by our atmosphere as a result of global warming. And this has caused a 1 degree increase in temperature so far. But the rate the oceans are warming is also increasing. The oceans are now warming at a speed that is 4.5 times faster than it was when I was in elementary school in the 1980s. Humans have also changed the oceans. As the oceans absorb carbon dioxide, they've become more acidic. And then in many places in the oceans, humans have also changed the balance of nature by removing top predators like sharks. But there's another threat to coastal habitats that many are probably less familiar with, and that's the effect of climate change on rainfall patterns. So warm air holds more water, so more water evaporates into warm air. And then warm air also, or warm water also supercharges hurricanes, giving them more energy. And so in this part of the world, one of the big effects we're feeling of climate change is that we're getting this increase in these big end of summer rainfall events. And these rainfall events can be a problem for animals that live in estuaries where rivers meet the ocean and saltwater and freshwater mix together. And one really important estuary species, and the subject of my lab's research, are eastern oysters. Oysters filter the water. They can filter the water at about 10 gallons per minute per oyster, or sorry, per day, per oyster. They provide habitat for other species because oyster reefs provide these three-dimensional structures with hiding places for baby fish. They help to prevent coastal erosion by helping to stabilize shorelines. And in Louisiana, oysters are an $80 million industry just in our state because oysters are tasty. Oysters prefer about a half and half mix of fresh water and salt water, and they actually get stressed out by too much fresh water. In 2019, the Louisiana Department of Wildlife and Fisheries documented up to 100% mortality death rates on some of the oyster reefs affected by this big high water event in the Mississippi that lasted through July that year. When temperatures are colder, oysters can survive freshwater for a shorter period of time, and that's because they just close up. But when they're closed, they're not breathing and they're not eating. So you can see in this picture on the bottom left of your screen, these are baskets of oysters that are the same age. They're all about 2 years old, and they're actually brothers and sisters. They were produced at the Louisiana Sea Grant Hatchery all at the same time. But the oysters on the left were raised at a high quality site with the right mix of saltwater and freshwater. The oysters on the right were raised at a site that had too much fresh water. And as a result, they're about half the size of their brothers and sisters that were grown in a better habitat. So the research we do in my lab is really aimed at understanding how oysters will respond to some of these stressful conditions. One of the things we're able to do is bring oysters from the wild back to the Sea Grant Hatchery in Grand Isle. And there we can breed them and raise them and test whether, for instance, some families or populations of oysters are more resilient to stress than others. We can also put juvenile oysters out in cages and put them out into different habitats and ask whether certain habitats are better for oyster growth and survival than others. Finally, we can expose oysters to different conditions in the lab and ask whether particular combinations of stressful conditions are worse than others. Some of the observations we've made: oysters from Louisiana are better at dealing with fresh water than oysters from Texas, but at least some oysters from Texas are better at dealing with low oxygen conditions than oysters from Louisiana. We've also found that certain combinations of stress are worse than single stresses experienced alone. For instance, warm temperatures and fresh water are probably especially bad for oysters. We think of them as being kind of like a bad drug interaction where they might be able to handle warming or fresh water on their own, but put them together and they're deadly. We hope that our research can help us to conserve and manage oysters. So for instance, knowing that some oysters are better at dealing with fresh water than others tells us we should choose the most resilient parents when breeding oysters for restoration projects. Knowing that fresh water is more stressful when the water is also warm tells us that the freshwater diversions planned for coastal protection in Louisiana will be less stressful for oysters if they're done when the water temperatures are cooler. So these are just a few examples of how science can be important to both management and conservation of important species. One more slide, sorry, thank you. So taking a step back, I guess I'd like to sort of finish my part of this conversation with some thoughts about the importance of both hope and optimism when thinking about conservation. Climate change can seem like a really enormous and overwhelming problem. And as a result, I think sometimes the reaction is just to sort of throw up our hands and think there's nothing we can do. And so I think it's important to remember some of the successes of previous conservation efforts. The pictures on the left are from the Bikini Atoll in the Marshall Islands. From 1946 to 1958, this was the site of 23 nuclear tests. And these nuclear tests released an energy equivalent to 76 megatons of TNT, resulting in widespread devastation to these coral reefs. But after that time, the Northern Marshall Islands were left relatively undisturbed by humans. And in a survey in 2002, 70% of the coral species had returned. It's not that they were exactly the same as they were before testing, the nuclear testing, but those coral reefs have experienced recovery. By the same token, the pictures on the right are showing you some examples of species who neared extinction, but whose populations have increased dramatically after protections were enacted. And so what these stories tell us is that conservation works whenever there's a will to enact protections. Finally, I want to say thank you to all of you. So much of the research we're going to talk about here this evening is funded by the National Science Foundation. And that means your tax dollars are doing this work. And so you should all give yourselves a pat on the back for supporting the science that helps to protect our oceans. And with that, I'm very happy to turn the stage over to Dr. Jacqueline Padilla-Gamiño. When I was told I could invite a guest to this event tonight, I knew immediately who I would most want to share the stage with. Dr. Padilla-Gamiño is a colleague and a good friend. And before we became professors, we both collaborated on research as postdocs at a lab in UC Santa Barbara. And the work we did there remains the hardest, but also some of the funnest research I've ever done. So please welcome Dr. Padilla-Gamiño.
Thank you so much. And yeah, it's a pleasure to be here at LSU Science Cafe and to have the opportunity to share the stage with Dr. Kelly, a great collaborator and friend. So today I'm going to talk about a little bit of what we do in my lab at University of Washington. And so my talk is organized in two different sections. The first one is the work that I do here in Washington in the Pacific Northwest, which is a temperate region. And then the other section of my work is actually performed in the tropical environment in Hawaii. So the work that I do at University of Washington in the Pacific Northwest centers around the future of seafood and really understanding what's going to happen to these organisms that are super important and have ecological and commercial value. And what can we do to make them more resilient and mitigate the effects of global change? Aquaculture is huge here in the Pacific Northwest. Shellfish along the West Coast is a 111 million industry supplying jobs for thousands of people in Oregon and Washington. And in my lab, we are working with some of the species that you see here on the slide. So we work with abalone, oysters, and mussels, and also purple hinge rock scallops. But also we work with seaweed, such as coralline algae, the pink seaweed that you see on the top right, and bulk kelp on the top left, which are going to provide the habitat for a lot of the organisms that we study. So it's understanding the habitat and the important commercial organism, it's just as important, because the success of one might depend on the other one. And so in my lab, we focus on ocean acidification experiments, temperature experiments, and anoxia experiments, which are conditions that might be found in the future for many of these organisms. In fact, the animals and plants or seaweeds in the Pacific Northwest are more vulnerable to ocean acidification, which is when the waters become more acidic and the organisms might have trouble calcifying, right? So in the Pacific Northwest, the waters are more vulnerable to ocean acidification because of two reasons. The first one is this upwelling that comes, so water from the deep that goes to shallow areas and brings a lot of nutrients, but also a lot of CO2, and makes them more acidic. And the other way in which the waters can become acidic here in the Pacific Northwest are actually by nutrient runoff. And so in the open coast, upwelling will be the main driver of ocean acidification. But in estuaries like the Puget Sound, then it's the input of rivers with a lot of nutrients that will be creating a lot of ocean acidification for many of these shellfish species. And so that's some of the work that we do. Next slide, please. And so one of the experiments that one of my graduate students is doing, Lindsay Alma, is we are collaborating with physical oceanographers that are putting these incredible orca buoys in different areas of Puget Sound and the open coast of the Washington coast. Yeah. And so these are amazing buoys because they are going to measure wind speed, light, temperature, salinity, oxygen, pH, right? And that's going to give us a really good understanding of what are the conditions experienced by the organisms. So we do lab experiments, but we are also doing these exciting new field experiments where we actually get to see the responses in situ, in the field of the organisms, in collaboration with oceanographers that are giving us a lot of information of what's happening in the water. And I think this is a very important link that sometimes is not very well studied, because most studies on ocean acidification are performed in laboratory conditions. And so on the right side, you can see one of my students. We did this PVC structure, and the black things are cages, and inside, that's where the shellfish are. And so every month, we will retrieve or grab different cages to measure different parameters or different things on the shellfish, like how much they grew, do they have lipids, is the strength, what is the strength of the shell? Because that will give us some insights on the environment as well. And so this is really important. Next slide, please. Because then what we do is we have cages at different depths. And then we get to see how organisms that are on the shallow areas compared to shellfish that are perhaps in deeper areas. And we'll do it throughout the year. So this is an example of shellfish in the winter. In the winter, the conditions are very different, because you see on the left how I have that is well mixed. That means that you see similar conditions because we have storms and wind. Then from 5 to 30 meters, the water is very well mixed. And next slide, please. And if you compare it to the spring, when we don't have as many storms or wind, you can see that it looks very different, right? The conditions on the top are very different from the conditions at the bottom. So we have a lot of different things happening, and our question is, how will the shellfish change as the conditions outside change? And this information is really, really important for shellfish growers. Next slide, please. Especially in the context of offshore aquaculture. And these long lines that sometimes are used to grow mussels or other shellfish, because organisms or shellfish on the shallower areas will have very different conditions with the ones compared to the bottom areas. And so this is really important because that can explain a lot of mortalities in different shellfish. And that can also give us a better idea of how to best mitigate against the effects of ocean acidification, of global warming. What can you do to your culture so that you have better shellfish that is more resilient and that is going to produce babies or offspring that are also going to be more resilient based on the conditions on the environment. So that's the type of work we do in the Pacific Northwest. Next slide. In the tropics is a little bit of a different story. When we go to the tropics, we go to the Hawaii Institute of Marine Biology, which is here, this picture on the top right. That's where there is a little island that has a super well, pretty big actually research center. And we work in Kaneohe Bay. And you see one of the picture on the top, on the bottom right. And the main question is how temperature is going to affect the corals. Next slide, please. And so temperature is a very important driver on the condition of organisms. On the left panel, you have a very healthy coral. It's brownish, it has beautiful color. And on the right side, you see the exact same coral after a bleaching event. And what is a bleaching event? A bleaching event occurs when the temperatures are really, really high. And then corals that have little symbionts or little seaweed or little plants inside of them basically get super stressed. And then they release this little seaweed into the water. And then they look white because they don't have these little plants that they have within their tissues anymore. And so what you're looking at is the skeleton. They're still alive, but they're kind of transparent and they look white because you're looking at the skeleton. And that's called coral bleaching. And corals that are bleached tend to be more vulnerable to stressors, to high temperatures, to ocean acidification. And so one of my questions is, what happens with reproduction? Are corals that bleach gonna be able to reproduce the same? Where are they gonna get the energy from? Because the little seaweeds that live within their tissues actually give them energy. And if they don't have them, where are they gonna get the energy to produce the eggs? Next slide, please. And so in my lab, we have a project that is NSF funded to try to understand that question. And so we grabbed different colonies, we broke them in half. And what we did is we bleached one colony and then we had the other one. And then we put them in these racks. Here, I am setting up some bleached colonies and some healthy colonies. And we left them there for a while, for a year, actually in the field. And then after a year, we went back and we tried to recover the colonies to see how much did they reproduce, how healthy they were, how quickly were they able to bring back their symbionts or their plants. Will the plants return to their tissues or did the corals die? And so that's the research that I'm doing right now. Next slide, please. And in addition to that, we return in a specific period of time in Hawaii when the corals actually release the eggs. So this is a little video that if we can start, it can show you how when the corals are ready to spawn, they spawn by releasing the eggs and the sperm into the water column. And so the eggs will go up because they have a lot of lipid, a lot of fat, and that's why they're all going up. And once they get to the surface, these eggs will find the sperm, fertilization will occur, and then you will have like baby corals that eventually will go back to the bottom of this ocean and settle and create another coral. And so what we did is we were able to catch some of the eggs of colonies that were bleached and didn't, and that were not bleached. And right now my students and I are doing a lot of lab work to try to see the differences between the eggs from the different types of corals, to try to better understand the effects of temperature. And with that, I would like to end. And yeah, I'm excited to hear your questions. Thank you.
Great. Thank you so much for those two talks. And we want to welcome the audience to type in any questions at the Q&A at the bottom of your screen. And to kick us off, what can we do as stewards of the environment, as people who enjoy the ocean, to help offset or even reverse the negative effects of climate change on our ocean? And maybe Dr. Kelly, can you kick it off?
Sure. So a couple things. I think the most important is probably to do everything that we can to try to slow climate change itself. And so we can do that through personal decisions to reduce the amount of fossil fuels that we individually consume, and also in advocating for policies that also help us to do that as a community. But then I think for the oceans, we also will need to be mindful of the fact that even if we stopped consuming fossil fuels today, the climate would continue to warm because of the fossil fuels that we've already used. And so there are going to be additional things that we need to do to protect species that have already been made vulnerable to climate change. And I think that's where Dr. Padilla-Gamiño's work really comes in, is in thinking about how warming might affect coral reproduction, for instance, and that can help us, for instance, to protect the species that we think might be most sensitive, either through supportive breeding programs or creation of reserves to try to boost the populations of the most sensitive species.
Yeah, I concur with Dr. Kelly, and also I want people to keep in mind that warming is just one of the many stressors in the ocean, right? So, so perhaps you cannot stop warming right away, but you can definitely advocate for better policies for water quality, for example, better waste treatment plants that are also contributing to the decline of many of the marine systems. So there's a lot we can do. And obviously, maybe you cannot change the whole CO2 story, but you can change other stories.
And we have a that's a good segue into a question from one of the audience members. Is it possible for climate change to cause oysters and other organisms in estuaries to become extinct?
So what we see in our research so far is that it's the effect of fresh water, that it, for Louisiana at least, that's the most important for oysters, although the combination of fresh water and warmer temperatures makes the effects of fresh water much more stressful. For many species in the ocean, what we think will tend to happen is that their whole populations will tend to shift. And so they may not go extinct, but they may move towards cooler places, places with more hospitable conditions in terms of the amount of fresh water. And so I think in our part of the coast, that's what we expect to happen in terms of the effect of fresh water, is that there will be winners and losers among the different oyster reefs. One of the reasons that's especially problematic in our part of the coast is that oystermen have leases that are particular locations in the Gulf of Mexico. And so if your particular oyster lease is impacted by fresh water, you can't necessarily get up and move to a better place for oysters. So that's a long answer to say that I don't think we expect them to become extinct, but we do expect them to move and to decline in some places and potentially increase in others. And that can sometimes have pretty important impacts for the humans that depend on them as well.
And here on the West Coast, I think they may not die because of temperature, but if there are a lot of nutrients in the water that might create anoxic conditions or conditions where oxygen is very low, that can definitely kill a lot of them. But I mean, maybe there will be other populations that will survive. But what we are seeing here in Washington is that for some species, the young individuals that we call larvae, right, tend to be more vulnerable to ocean acidification. And so some of those species, for the young ones, they might have trouble calcifying or producing their shell. And that might have some consequences also in their distribution and their abundance.
Dr. Padilla-Gamiño, here's a question from Hassan Zaman. He was wondering, was there any change in the lipid profile of the shellfish that you harvested? And if yes, which type of lipid?
So the fatty acids data, I know we have it. I'm just not super familiar, but we definitely, what I can say is that we found a higher lipid content in the shellfish that were in the shallower areas, for sure. And we do have fatty acid profiles, but I wasn't expecting such a sophisticated question. If you can contact me, definitely I can let you know what type of fatty acids are we finding for the species at different depths. Yeah, I'm more than happy to share that information.
Dr. Kylie, this is a question from Chaya and Anjali Chakrabarti. They're wondering, why are the oysters smaller than when closer to fresh water than pure marine habitat?
It's a great question. Hi, Chaya and Anjali. Great question. So two things. First, when they get really stressed out by fresh water, they close up their shells to kind of protect themselves from the fresh water. And so then they stop eating. So one of the effects is that when they're really stressed out and they stop eating, they don't have enough energy to grow. But probably the other thing that happens is that the salt water also has more calcium in it, and the oysters need that calcium to grow their shells. And so the oysters that are closer to fresh water probably don't get enough calcium to grow their shells as well. Great question.
Okay, I have a couple questions about the filter feeding abilities of oysters. Do you believe that oysters who have experienced genetic mutations and are resistant to acidity can purify the water in certain bodies of water? And what's your opinion on this? And then this might be another related sort of question. I've read that oysters and possibly mussels do a great job to filter the water. My question is, what happens to the bad stuff that's getting filtered out? Does it remain within the flesh of those shellfish?
Let's see, maybe I'll try to tackle the first question first. So, and I think Dr. Padilla-Gamiño could probably speak more to the effects of acidity. Our focus has been on variation in their ability to deal with low salinity with fresh water. And we do see that oysters from Louisiana are better at dealing with fresh water than oysters from Texas. And so I would guess that the ones that are better at dealing with fresh water are also going to be better at filtering the water because they're less stressed out than the ones that are really stressed out by that fresh water and staying closed all the time. And then maybe can I hand it over to you, Dr. Padilla-Gamiño, to talk a little bit about acidity?
Yeah, actually, I wanted to talk about what happened to the stuff, right? Once it's filtered, and different things can happen because bivalves are unbelievable, right? Like you just look at them like two little shells, but actually they have a lot of filtration mechanisms. So, for example, another thing that I study are actually microplastics. And so microplastics that are in the water, right? Just like other stuff that is in the water. And so one of our hypotheses was like, okay, you have the microplastics and we have the oysters, and we know that the oysters filter a lot of water. Are we gonna find just a bunch of microplastics inside the oysters, right? So if you eat an oyster, are you actually eating a bunch of microplastics? So that was our question for Washington. And so one of my postdoctoral researchers, Julieta Martinelli, actually went all over, grew up Pacific oysters from a lot of places. Then we took the tissue out, we put some chemicals, we extracted, and we were looking for the plastics. And we found not a lot of them because actually the oysters have a very high capacity to filter a lot of things that they don't like. And they have multiple mechanisms. And they might get it in, but they immediately like release it. And so it depends on whether the oysters can use whatever they filter. They have very sophisticated strategies to actually keep it and use it, or just release it. And they can release it as what we call poop, feces, or they can release it as what we call pseudo-feces, which is like pseudo-poop, like kind of poop that is not a real poop. And so some things that will be filtered by the oysters might stay in the oysters, be used by the oysters for growth, and some they might just reject and put in the sediment, where maybe a sea cucumber will come in and grab it. Who knows? So, but that's a really interesting question about, energy, right? Where do things go? What happens? Who takes them? Yeah, good question.
Another question, Dr. Padilla-Gamiño, about coral. Dr. Licata is wondering, which hurts coral more, rising temperature or acidification? Temperature.
If the corals die, will die because of temperature, not ocean acidification. I mean, if you don't have temperature and just ocean acidification, maybe after a long period, they might die, right? But it's temperature. Temperature is what sets the stage for the stress. Like temperature is the worst enemy right now for corals. And they are having a really bad time worldwide. In 2015 through 2017, we had major bleaching events around the world. And in some areas, like the Great Barrier Reef in Australia, around 30% of the coral colonies there died. It's pretty severe, so temperature.
you. James Madden has a question. I think, Dr. Kelly, if sea level rises, the coastline will change, especially in Louisiana. Will some oyster reefs, for instance, shift towards this new coast and away from their current geographic location? Would they move?
Absolutely. Both for changes in where the optimal salinity conditions are, and changes in where the places are that they can settle. Oysters have a larva that is just swims around in the water for about 2 weeks before they settle. And so that means that if they have a place that they can settle, oysters can colonize a new habitat each generation. But one of the things they do need, in addition to the right salinity conditions, is something hard to settle on. So often that is in the form of other oysters. And that is one of the challenges to settling in a new place, is if you're the first oyster larva there and you just have soft mud, you have nothing to land on. But once they start to get established, they can build up a population in a new area.
Okay. Also, so we have another question. To what degree do fisheries managers, aquaculturists, conservationists need to consider preserving genetic diversity in combating the effects of climate change?
That's an excellent question. And I think, yes, that's an important part of the story. That's part of why we're interested in knowing whether oysters from different places in the Gulf of Mexico can deal with different environmental conditions. Is it might be that if you're concerned about the effects of particular changes, you want to conserve that genetic diversity, that ability to deal with those future conditions.
Yeah, for corals, work is the same. And that's why we try to bring the egg and the sperm to have this genetic diversity. Because in some areas, they restore coral reefs by just cutting a big colony and replanting those fragments, but they have the exact same genetic identity. But by grabbing egg and sperm, you can make, new offspring that is genetically diverse. So you can have asexual or sexual type of coral restoration efforts. And both of them are important. And they have their advantages and disadvantages.
Might be a follow-up to that. So what different breeding conditions are necessary for an oyster or coral population to become more resilient to stress caused by climate change and other factors? So the breeding conditions, I guess.
Do you want to speak to that with corals?
Sure. So sometimes, for example, when you decide to do these fertilization experiments or in vitro experiments with corals, right, you need to decide, well, where am I going to get the parents from? And generally, you want to choose parents that, for example, are in areas that have survived high temperatures, or you want to find parents that potentially live in areas with a lot of environmental variability, right? Where the environment, the water change a lot. Because if they are able to live in those conditions, then the hypothesis is that the offspring might acquire part of that ability and also become very resilient. So that's one of the things that people do in coral restoration. Try to find like good mamas, good papas that, are pretty resilient and hopefully will have resilient offspring as well.
I can speak to that a little bit with oysters as well. Similarly, so my student Joanna Griffiths, who has now graduated, did a project where she compared the offspring of oysters that had lived under different conditions. And for oysters, she actually found that the parents from the most stressful site, they had just experienced those more stressful conditions their whole lives, when their offspring were moved to good conditions, those were actually the offspring that did the very best. It was as if they had been starving for good conditions, and they were ready to take advantage of them. And so what that showed is that there's the genetic diversity of the parents matters, but also just the conditions that the parents had experienced changed how resilient their offspring were as well.
I have another question about ocean acidification. At the beginning of your talk, you mentioned that it's hurting organisms, and Eva is wondering if ocean acidification is unavoidable because there will always be carbon dioxide.
Do you want to speak to that, Dr. Padilla-Gamiño?
Well, so I guess in the environment, there might be environments that naturally they are more acidic, right? Like upwelling, like upwelling areas where deep waters come up to the surface and have a lot of nutrients. They have a lot of CO2 and have low pH. But the idea here is, if we are able to decrease the amount of CO2 that we are releasing, the idea is that there will be, the problem right now with CO2 is that, and with temperature, is that it's increasing at rates and speeds, right, that we did not see before. So it's not just that it's more, but that is, we're getting faster. CO2 is increasing in a very extreme way, and is creating very different conditions in other places. So I don't think that, I think we can definitely reduce ocean acidification by reducing CO2 in the environment. But there will still be always some areas that are naturally more acidic than others. And maybe Morgan can complement. Yeah.
Yes, I agree with everything you just said, that there will always be CO2, but we still have a choice about how fast it increases, and it's the speed of the increase that is especially stressful.
Mm-hmm. Also because organisms need some time, right, to really deal with those changes. And that's part of the problem now. It's not that organisms cannot deal, but the rate of change compared to the rate that we are producing the change is different. And that's what the problem is.
So Dr. Kelly, we have a question from VJ. He's wondering what happens when oysters are born in waters that have, what happens when oysters are born in waters that have high acidity? Do they experience a gene that can be resistant to acidity or not? Is this process the same or different?
You know, we have not directly tested the effects of acidity on oyster larvae. Dr. Padilla-Gamiño, have you done experiments to test acidity on oyster larvae?
No, what we have is oysters that come from acidic environments, which is kind of testing. So what we had are the parents that we put in more or less acidic conditions. And then we grab egg and sperm from those parents. We made some new baby oysters. And what we do find is that they have different sensitivities depending on the conditions of where the parents were. So yeah, we have some of that data, and sometimes it's not as clear, right? So sometimes you have offspring or baby oysters that even though they were from parents that were in very different conditions, the offspring actually shows very similar conditions. to other larvae that had very different parents. So I don't know, the more I study in the field things, I think sometimes the more difficult it is to really distinguish what's causing what, because there are so many things acting at the same time, right? The feeding, the light, the temperature, the pH, the hormones, right? Released by medicine. I mean, there's so much stuff. There's so much stuff in the water. And we still need a long way to go to really see how all those players interact and affect the response of something.
I can offer a related answer also related to salinity about changes in genes. So this is for freshwater, not for acidity, but also my student, now Dr. Joanna Griffiths, did another experiment where she took a bunch of oyster larvae and she exposed them to fresh water, and she kept just the ones that survived, and she sequenced their DNA to compare the DNA of the baby, the oyster larvae that survived, to the ones that had died. And she did find that the ones that survived, they had particular versions of genes that seemed to make them better at being able to survive the fresh water than the ones that had died. So you might be able to do a similar experiment with acidity.
Here's a question for you, Dr. Padilla-Gamiño. Henry Chen is wondering, are you breeding the coral in your project, or using selective breeding and picking the species that look to be the most damaged versus the healthier population? I guess, what type of coral species are you studying in terms of the bleaching project?
Right now, I'm working with Montipora capitata, which is a coral from the family Acroporidae, genus Acropora, and it's called rice coral because it has like little bumps throughout the body, and this coral is a hermaphrodite spawner. That means that the coral will produce both egg and sperm at the same time, and that it will release. So those little like spheres that you saw in the video is actually an egg sperm bundle. So it's a little package that contains both egg and sperm. And I'm working with that species because it's the most abundant species in Kaneohe Bay in Hawaii. It's a very important reef builder, and it's beautiful to see and to work with. Yeah, it creates beautiful ecosystems for other fish and other organisms to live in.
Here's a question from Dr. Kyle Harms. He says, my limited understanding is that noise pollution is increasingly recognized as a human-caused environmental change to marine organisms. Examples are often given about difficulties faced by whales, but I wonder whether shellfish are affected. Is noise a stressor also for shellfish?
Mm-hmm.
You know, I can't give you an answer right now, but I know where to look. There's a group of researchers at Northeastern University who I know are researching the general effect of sound on oyster reefs. Both, I believe, looking at whether oysters tend to close when they hear sound, but also whether predators and prey hide more or less in the presence of sound. Yes, so I can't give an answer, but I do know that is something that other folks are working on. Dr. Padilla-Gamiño, do you have anything to add for the shellfish you work on?
No, not the shellfish, but the corals. So there are some studies that coral larvae may recognize the reef and where to settle based on sounds. So there are some studies that have shown some evidence on that.
Peg Riley would like to know, what are the factors that increase oyster vulnerability to bacterial and viral threats? We've heard of elevated risks in both Louisiana and Puget Sound waters.
That's a great question. And that's a very sort of active area of research. So a student who was an undergraduate in my lab, who's now a PhD student in Rhode Island, Megan Guidry, was looking at the microbiome of oysters, looking at their whole community of bacteria. And one of our questions was whether oysters with a particular community of bacteria might be more resistant to a disease that oysters get called dermo. So that's sort of an ongoing analysis in our lab. I know that others are also interested in trying to understand whether you could actually give oyster larvae being cultured in a hatchery, could you give them a sort of a probiotic that helped to maintain a healthy community that might then help to prevent against disease?
Yeah. And I think it's important to think about what happens when you get sick. Right? You get sick because maybe you did not sleep well, you did not eat well, right? So similar, or maybe you are stressed. So similar things happen to marine organisms, right? If they have crazy temperatures they can deal with, or they have to do a lot of extra effort to cope with what's around them in the water, then they might be more vulnerable to disease.
So we have so many great questions. It's been a really great discussion. I have one last question I really would love to throw to you. It's from Madeline Kelly. She wants to know, what can we do to encourage younger students who feel powerless because they cannot yet vote to still advocate for policies to fight climate change?
That is coming from Madeline Kelly, who's a really, I happen to know, really excellent middle school teacher in Baton Rouge. I think that even students who can't vote yet have a powerful voice with all of the adults who do vote that they can talk to, and they can write letters and advocate with the adult leaders that they know to try to change minds for the issues they feel passionate about. And it helps to have teachers providing them with good ammunition and facts as well.
Yeah, and time goes by very quickly. So soon enough, you'll be able to do it and to take decisions and influence others. So just wait, don't lose that feeling, don't lose it, and then take action when you can.
Great. Well, thank you so much for this wonderful presentation and great discussion. We really appreciate you, and look forward to continuing to follow your research and yeah, and continue the dialogue as you know, our world is changing. I also want to share with everyone, we have a lucky winner for tonight's WRKF drawing is Mamie Broadhurst, and WRKF will mail you the Science Friday beverage tumbler. We really look forward to seeing everyone again next month. Tuesday, March 30th, it's going to be with Dr. Theda Daniels-Race and Dr. Paul Race. They're going to be giving an engaging talk on nanoscience and talking about the science and ethics of the very super small. So we look forward to seeing you then, and thanks so much for joining us.